The study of nucleic acids began with the discovery of DNA, progressed to the study of genes and small fragments, and now exploded to the field of genomics. Genomics is the study of entire genomes, including complete sets of genes, their nucleotide sequence and organization, and their interactions within species and with other species. DNA sequencing technology has contributed to the advances in genomics, just as information technology has led to Google Maps that enable people to obtain detailed information about locations around the globe. Researchers use genomic information to create similar DNA maps of different organisms. These findings have helped anthropologists to better understand human migration and have aided the medical field through mapping human genetic diseases. Genomic information can contribute to scientific understanding in various ways, and knowledge in the field is growing quickly. Biotechnology is the use of biological agents for technical advancement. Biotechnology was used for breeding livestock and crops long before people understood the scientific basis for these techniques. Since the discovery of the structure of DNA in 1953, the biotechnology field has grown rapidly through both academic research and private companies. The primary applications of this technology are in medicine, vaccine and antibiotic production, and agriculture, crop genetic modification in order to increase yields. Biotechnology has also many industrial applications such as fermentation, treating oil spills, and producing biofuels. To study and manipulate nucleic acids, one must first isolate or extract DNA or RNA from the cells. Researchers use various techniques to extract different types of DNA. Most nucleic acid extraction techniques involve steps that break open the cell and use enzymatic reactions to destroy all macromolecules that are not desired. A lysis buffer, a solution which is mostly detergent, breaks down cells. Note that lysis means to split. These enzymes break apart lipid molecules in the cell membrane and the nuclear membranes. Enzymes such as proteases that break down proteins and activate macromolecules, and RNAs, ribonucleases, break down RNA. Using alcohol precipitates the DNA. Human genomic DNA is usually visible as a gelatinous white mass. One can store the DNA samples frozen at -80 for several years. Because nucleic acids are negatively charged ions at neutral or basic pH in an aqueous solution, an electric field can mobilize them. Gel electrophoresis is a technique that scientists use to separate molecules based on size using this charge. One can separate the nucleic acids as whole chromosomes or fragments. Nucleic acids load into a slot near the semi-solid porous gel matrix negative electrode and pulled toward the positive electrode at the gel's opposite end. Smaller molecules move through the gel pores faster than larger molecules. This difference in migration rate separates the fragments on the basis of size. There are molecular weight standard samples that researchers can run alongside the molecules to provide a size comparison. We can observe nucleic acids in a gel using various fluorescent or colored dyes. Distinct nucleic acid fragments appear as bands at specific distances from the gel's top, the negative electrode end, on the basis of their size. Although genomic DNA is visible to the naked eye when it's extracted in bulk, DNA analysis often requires focusing on one or more specific genome regions. Polymerase chain reaction, PCR, is a technique that scientists use to amplify specific DNA regions for further analysis. Researchers use PCR for many purposes in laboratories, such as cloning gene fragments to analyze genetic diseases, identifying contaminant foreign DNA in a sample, and amplifying DNA for sequencing. More practical applications include determining paternity and detecting genetic disease. Scientists can probe nucleic acid samples, such as fragmented genomic DNA and RNA extracts, for the presence of certain sequences. Scientists design and label short DNA fragments called probes with radioactive or fluorescent dyes to aid detection. Gel electrophoresis separates the nucleic fragments according to their size. Scientists then transfer the fragments from the gel onto a nylon membrane in a procedure we call blotting. Scientists can then probe nucleic acid fragments that are bound to the membrane surface with specific radioactively and fluorescently labeled probe sequences. When scientists transfer DNA to the nylon, they refer to the technique as southern blotting. When they transfer RNA to a nylon membrane, they call it northern blotting. Scientists use southern blots to detect presence of certain DNA sequences in a given genome, and northern blots to detect gene expression. In general, the word cloning means the creation of perfect replica. However, in biology, the recreation of whole organisms is referred to as reproductive cloning. Long before attempts were made to clone an entire organism, researchers learned how to produce desired regions or fragments of the genome, a process that is referred to as molecular cloning. Cloning small genome fragments allows researchers to manipulate and study specific genes and their protein products, or non-coding regions in isolation. A plasmid, or vector, is a small circular DNA molecule that replicates independently of the chromosomal DNA. In cloning, scientists can use the plasmid molecules to provide a folder in which to insert a desired DNA fragment. Plasmids are usually introduced into bacterial hosts for proliferation. The bacterial context scientists call the DNA fragment from the human genome foreign DNA or transgene to differentiate it from the bacteria's DNA or the host DNA. Plasmids with foreign DNA inserted into them are called recombinant DNA molecules because they are created artificially and do not occur in nature. They're also called chimeric molecules because the origin of different molecular parts of the molecules can be traced back to different species of biological organisms or even to chemical synthesis. We call proteins that are expressed from recombinant DNA molecules recombinant proteins. Not all recombinant plasmids are capable of expressing genes. The recombinant DNA may need to move into a different vector or host that is better designed for gene expression. Scientists may also engineer plasmids to express proteins only when certain environmental factors stimulate them, so they can control the recombinant protein expression. Reproductive cloning is a method scientists use to clone or identically copy an entire multicellular organism. Most multicellular organisms undergo reproductive by sexual means, which involves genetic hybridization of two individuals, parents, making it impossible to generate an identical copy or clone of either parent. Recent advances in biotechnology have made it possible to artificially induce mammal asexual reproduction in the laboratory. Genetic engineering is the alteration of an organism's genotype using recombinant DNA technology to modify an organism's DNA to achieve desirable traits. The addition of foreign DNA in the form of recombinant DNA vectors generated by molecular cloning is the most common method of genetic engineering. The organism that receives the recombinant DNA is a genetically modified organism, GMO. If the foreign DNA comes from a different species, the host organism is transgenic. Scientists have genetically modified bacteria, plants, and animals since the early 70s for academic, medical, agricultural, and industrial purposes. In the U.S., GMOs such as Roundup Ready soybeans and boar-resistant corn are part of many common processed foods. Scientists call the process of testing for suspected genetic defects before administering treatment genetic diagnosis by genetic testing. Depending on the inheritance patterns of disease-causing gene, family members may be advised to undergo genetic testing. For example, doctors usually advise women diagnosed with breast cancer to have a biopsy so that the medical team can determine the genetic basis of cancer development. Doctors base treatment plans on genetic test findings that determine the type of cancer. If inherited gene mutations cause the cancer, doctors also advise other female relatives to undergo genetic testing and periodic screening for breast cancer. Doctors also offer genetic tests for fetuses or embryos with in vitro fertilization to determine the presence or absence of disease-causing gene in families with specific debilitating diseases. Gene therapy is a genetic engineering technique used to cure disease. In its simplest form, it involves the introduction of a good gene at a random location in the genome to aid the cure of a disease caused by a mutated gene. The good gene is usually introduced into disease cells as part of a vector transmitted by a virus that can infect the host cell and deliver the foreign DNA. More advanced forms of gene therapy try to correct the mutation at the original site in the genome, such as is the case with the treatment of severe combined immunodeficiency. Although several recombinant proteins in medicine are successfully produced in bacteria, some proteins require a eukaryotic animal host to properly process it. For this reason, the desired genes are cloned and expressed in animals, such as sheep, goats, chicken, mice. We call animals that have been modified to express recombinant DNA transgenic animals. Several human proteins are expressed in transgenic sheep and goat milk, and some are expressed in chicken eggs. Scientists have used mice extensively for expressing and studying recombinant gene and mutation effects. Manipulating the DNA of plants, i.e. creating GMOs, has helped to create desirable traits such as disease resistance, herbicide and pest resistance, better nutritional value, better shelf life. Plants are the most important source of food for the human population. Farmers developed ways to select for plants varieties with desirable traits long before modern day biotechnology practices were established. We call plants that have received recombinant DNA from other species transgenic plants because they are not natural. Government agencies closely monitor transgenic plants and other GMOs to ensure that they are fit for human consumption and do not endanger other plant or animal life. Because foreign genes can spread through species and environment, extensive testing is required to ensure ecological stability. Staples like corn, potatoes, tomatoes were the first crops that scientists genetically engineered.